Hello students, hope you will be fine at your native places. Last time we were discussing this process of pollination and we have discussed various types of the pollination, that is the self-pollination and the cross-pollination. And during this process of pollination, what we have studied that how the male gametes, how the pollen tubes, they are taken to the female reproductive part or to the stigma. The process of fertilization in angiosperm or in the flowering plants, it was first of all discovered by the Strasburger in the year 1884. Very good. After this, what we will study that now what we will study that the pollen tube जो हमारे पास है after pollination, the pollen grains forms the pollen tube. After pollination, what we will see that the pollen grains, they form the pollen tubes, and this process is called as siphonogamy. What we can discuss is that it is the formation of pollen tubes. Now, the formation of this pollen tube, it is stimulated by the sugary substance secreted by the stigma. This pollen tube, it contains the two, along with the two pollen cells, as two pollen cells of pollen game and one tube nucleus. It, what we'll see, it mainly reaches to the ovule where it has to release these pollen, what we call as these pollen cells. Now, the length of the pollen tube, it mainly depends upon the length of the style. Depending upon the internal structure of the style, we have mainly got two types of the styles. एक को हम बोलते हैं hollow style. Second one जो हमारे पास है, that is called as the solid style. Hollow style जो है, it is a wide canal which is lined by the canal cells. Wide canal. And in this, instead of this canal cells, we have got the conductive tissue. Now, how this pollen tube, when it reaches this ovule, now it makes its way into the inside the ovule, and there are the three different ways by which this pollen tube it may enter inside this ovule. First one, when it enters this pollen, when it enters through the micropylar end, that it is called as the progamy. When it enters, and this progamy, it is rather most common in the angiosperm, that the pollen tube it enters into this embryo sac through the micropylar end. When this pollen tube it enters through the base, or we can call it as the chalaza, then it is called as the chalazogamy. When it enters through the integuments, it is called as mesogamy. On piercing, जब यहाँ की ओर चला जाए, when it pierces through these nuclei, when it piercing the nucleus, the pollen tube, हम कहते हैं it penetrates this embryo sac to release the two sperms in the megagametophyte, or what we call in the embryo sac. The cells of an unfertilized ovule, जो हम यहाँ पर होते हैं, they are total 8 in number, and they are arranged in 3 plus 2 plus 3 arrangement. These cells which are present in this embryo sac, they are arranged in 3 plus 2 plus 3 from top to bottom. The first three, they are antipodal cells, like this. They are antipodal cells. These two nuclei here, they are the polar nuclei, and there are the three, 1 2 3. दो में से हम क्या कहते हैं, जो these two, they are the polar cells या polar nuclei. Then these three, they have been further arrangement into 2 plus 1. 2 are what, this two जो यहाँ पर है, they are the synergids. and this one is the egg cell. This is the egg cell. This there are total 8 cells. 8 cells यहाँ पर है. These 3 are, हम देखेंगे जो इनका अरेंजमेंट है 3 + 2 + 3. These 3 they are called as the antipodal cells, जो हमने ऊपर की ओर देखे. These 2 they are the polar nuclei. There are again 3, 1 2 3. The 2 are the synergids and the 1 is the egg cell. Now when this pollen tube it enters into this embryo sac, where it has to release its what we call as these pollen cells. One pollen cell it fuses with this egg cell. One pollen cell it fuses with the egg cell, forming a zygote which is diploid. One pollen जो हमारे पास है, एक pollen cell it fuses with this egg cell. ये किसके साथ? egg cell के साथ fuse कर लेता है, as a result of which diploid zygote is formed, and this process is called as syngamy. अब जो दूसरा हमारे पास है, दूसरा pollen nucleus, it reacts, it fuses with these 2 polar nucleus and it forms a triploid nucleus. It forms a triploid nucleus which is called as primary endosperm nucleus, or which is simply called as PEN, which is called as PEN, which later on gives rise to the endosperm. Thus the process of fertilization which occurs twice, अब जो यहाँ पर process of fertilization होती है, that occurs twice in the same embryo sac at a time, that is called as double fertilization. यहाँ पर हम देखते हैं कि एक पोलन सेल वो एग सेल के साथ, और दूसरा जो पोलन जो न्यूक्लियस होता है it reacts with these 2 polar nuclei. In this case we will see the 2 fertilizations takes place, and this process is called a double fertilization. And this process of double fertilization, it was first of all discovered by S.G. Navician in the year 1897 in the lily species. This whole process. Now इसके बाद हम क्या देखते हैं, द ओवरी जो होती है which surrounds the ovule, it develops into the fruit and it is having the seeds which may penetrate, which may function, and it may function to disperse them. जो zygote यहाँ पर होता है, जो zygote form, it has got the full complements of the chromosome, that it is diploid in nature, while as the endosperm nuclei जो होता है, it has got 3 chromosomes. कितने होते हैं? 3 chromosomes, that it is triploid. The endosperm nuclei divides mitotically to form the, जो ये क्या बनाता है, it forms the endosperm, and this endosperm जो होता है, it acts as a nutritive tissue for the developing embryo and the subsequent germinating seeds. Now there are some significance, or we can say the importance of the double fertilization. First one, जो double fertilization हम देखते हैं, first significance इसमें है that it involves both the male gametes produced by the pollen grain. First significance, अब pollen grains में 2 pollen ये होते हैं male gametes, they both are being consumed, they both are being used by the pollen grain. Second, जो second fertilization product यहाँ पर होता है, that is this endosperm, this primary endosperm nucleus, it develops into the endosperm, which is a nutritive tissue, and this nutritive tissue it provides nourishment to the developing embryo. That is all about today. Rest, okay, stay home, and next time we will proceed further. Thank you, beta.